Hey, so today I want to take some time to kind of lecture a little bit and get through something that we would do on the board anyway. And maybe we can knock this out in a short little 8 to 10 minute video, and you'll have this in your notes, and we can kind of proceed in class, okay? So what I want to do is, we sketched out the first stage of meiosis the other day, called prophase 1. And we also talked about the differences in mitosis and meiosis. So this is going to be the 8 stages of meiosis, right? Why are there 8 stages? Because there's 2 divisions. So we said last Friday there's meiosis 1 and meiosis 2. And so I want to kind of cover that, okay? And I've just got some pictures of each phase, okay? And what I'll ask you to do is pause my video and do your best to sketch this out. I will explain what's happening in each one, okay? And we'll see how it goes, all right? So here, the very first stage we sketched in our notes the other day, and we have a nucleus that's going to break down, okay? And we have our spindle that's going to form, right? Centrioles and spindle fibers. But we talked about a process called crossing over. And that's where these homologous pairs, okay, of chromosomes, I've got 3 homologous pairs. These 3 homologous pairs are going to group up, sort of intertwine, and actually exchange different sections of these chromosomes, and that leads to some genetic diversity in our sex cells, okay? Remember that only cells in your body that are going to go through these 8 stages are your sex cells. We call those your gametes, okay? Not your somatic cells like skin and muscle, okay? So go ahead and pause this for a second, and I want you to do your best to sketch out prophase 1 if you didn't already, okay? And then any labels and anything that's described at the bottom, do the same thing, okay? Get that written down. So my video won't be very long, but I'm going to progress through each one of these, and you'll have to pause as we go, okay? All right, so there's prophase 1. Now, we move into metaphase 1, and this sort of looks just like mitosis, but what's different is that the homologous pairs are lined up across from each other, okay? That is not a single chromosome right here. That is like a blue chromosome from, let's say, your dad, and a red chromosome from your mom. Those homologous pairs are lined up in some random way across from each other on that equator, that middle line, okay? So pairs of homologous chromosomes move to the equator of the cell, and they do so, like I said, in pairs, all right? So it's a little different than mitosis because of how they're arranged on the equator, all right? So go ahead and pause it and sketch what that looks like. It's a great picture of these homologous pairs getting pulled apart. So instead of a duplicated chromosome getting pulled apart, we have an entire homologous, one of each one getting pulled to the pole. And I'm going to zoom in here for a second so you can kind of see we've got some color differences, right? And that's what took place in crossing over. I kind of showed that where we exchanged gloves with a couple of volunteers in class the other day. We exchanged like gloves and shoes, okay? So the chromosomes, as they get pulled to the poles, right? We're pulling a duplicated chromosome apart, okay? So this duplicated chromosome is going to this side, and its homologous partner is going to the other side, okay? So imagine if I was sketching this, it would look like a kind of like a big X getting pulled apart. Matter of fact, I have some sketches, and I did this. It would almost look a little bit more like that. Okay, the full X getting pulled apart, okay? So, back to anaphase 1, all right? You can pause that, take the notes on it. That's anaphase 1. And then finally, telophase and cytokinesis. So what do I have on either side here? I have duplicated chromosomes and one from each homologous set, okay? One from each homologous pair, okay? And that's the very first division. So what did I do during that division? I took what would have been a total of 46 chromosomes in the beginning, and I'm putting 23 duplicated chromosomes in each cell, okay? So in terms of how many chromosomes in a human, 46 to 23, 46 is what's called our diploid number, di standing for 2, and 23 in each now, that's our haploid number, or half the number of chromosomes, okay? So now I have two cells. So each drawing I'm going to have 2 cells as I move into meiosis 2. Now meiosis 2 is going to take our duplicated chromosomes and it's going to split those in half. Okay? So I get a brand new spindle. I've got duplicated chromosomes in each cell, and this has to go through the phases pro, meta, anatella to split the duplicated chromosomes in half. So as you watch this, ready? I'm going to you should pause and sketch, but I'm going to progress through this. It's actually going to look just like mitosis. Look, that looks just like mitosis. So what am I doing differently here? Is I've got 2 cells where a duplicated chromosome is going to get split into 2. All right? Just like mitosis almost, right? Taking these duplicated chromosomes and these sister chromatids are getting pulled to each side. And here's your end result. Your end result is 1, 2, 3, 4, 4 cells, okay? And each one has basically one copy of every gene or one copy of every chromosome in that cell, okay? So now with just another minute or two, let's just kind of talk about kind of the end of the line, like what did we do in this process? And that's how I'll end today, okay? So the process itself started off with a single cell with a nucleus and 46, in a human 46, right? Who knows? A carrot could have 12, a diploid number of 12 chromosomes in its somatic cells, okay? Somatic cells are the non-sex cells. It could have a number of 12. Well, what would a carrot need to do by the end of the steps in meiosis? It would want to cut that number from 12 down to 6. In a human, we're going to cut it from 46 down to 23. Okay, so that an egg has 23 chromosomes and a sperm has 23 chromosomes, and when they come together during fertilization, we make 46. Okay, so ultimately we want to go from 46 chromosomes, 46 duplicated chromosomes, and we want to divide that into 2 cells, each with 23 duplicated chromosomes. Okay, sorry for the shorthand. That's duplicated. And then through meiosis 2, we're going to take these guys and divide them up with 23 unduplicated chromosomes, or basically what you'll have here is those sister chromatids in each one. 23 sister chromatids in each one. Okay? So what did you do in a male? Well, it's easy. Need a new piece of paper. In a male, you took one cell and you turn that into 4 viable, meaning they all work, 4 viable sperm cells. Okay? This first cell, it has a name. It's called a germ cell. Okay? So it's not a sperm cell turning into 4 viable sperm cells. It's a cell in your body. It's in your reproductive area, and it's called a germ cell. All right. In females, girls, what they do is they have again a germ cell. This germ cell goes through 2 divisions, just like the males. Okay. The result's a little different, however. The cytoplasm doesn't split evenly. So in females, they only produce 1 egg, and then these 3 remaining smaller cells, they have a name. They're called polar bodies. They're basically non-viable. They don't work. They're not going to work as an egg. Okay? Non-viable. They don't. They're not going to be fertilized. They're basically, I guess, waste. Okay? So girls, you produce 1 egg from your germ cells, and boys, our germ cells can produce 4 working or viable sperm cells. Okay? Each is haploid. It has half the number of chromosomes in it. Okay? This is a haploid egg. Okay? All right, so that's meiosis, the production of your sperm and egg cells, and sketches to go along with each one. Okay? All righty, that's it.